Before mitosis can occur, the cell must first go through interphase. Interphase includes 3 phases: G1, S, G2. Interphase begins with the G1 phase. The cell will be the smallest it's ever been in this phase. Next comes the S phase, which main activity is to replicate the DNA of the cell, the chromosomes. After the chromosomes are replicated, the cell enters the next growth phase, G2. During G2, the cell grows and prepares for mitosis. Mitosis has 4 stages: prophase, metaphase, anaphase, and telophase. Mitosis begins with prophase. In early prophase, the chromatin fibers condense to form chromosomes inside a nuclear envelope. Next, the nuclear envelope disintegrates and nucleoli appear. Spindle fibers begin to form. These spindles move outward from the centrosomes. The centromere of each chromosome attaches to the spindle. Then, the centrosomes move towards opposite poles of the cell. Mitosis' next phase is metaphase. The chromosomes move to the middle of the cell. This is also called the metaphase plate. The centromeres lie on this plate. The centrosomes are now at opposite poles of the cell. Next comes anaphase. It begins when the sister chromatids of each chromosome are split. The chromosomes move towards opposite ends of the cell. This movement is caused by the shortening microtubules of the spindle. The centromeres are attached to the microtubules, so they move towards the poles first. At the end of anaphase, each pole has a complete identical set of chromosomes. The final phase of mitosis is telophase. The nuclear membrane begins to reform around each set of chromosomes. The chromosomes decondense to form chromatin. The nucleoli reappear and the spindle apparatus disappears. The cell is now ready for cytokinesis. Cytokinesis in animal cells involves the inward pinching of the plasma membrane to form cleavage furrows. In plant cells, cytokinesis forms a cell plate. Both processes result in daughter cells that are genetically identical. While mitosis results in identical cells, meiosis results in sex cells. These cells only have half the amount of chromosomes that a normal cell has. Meiosis begins with prophase 1. The DNA condenses and the chromosomes become visible. Homologous chromosomes are attracted to each other and pair up. One from the organism's father, another from the mother. Crossing over occurs. Crossing over allows DNA from maternal and paternal chromosomes to mix. Then the spindle fibers form. During metaphase 1, the homologous chromosomes line up at the cell's equator. Next, the nuclear membrane disintegrates. During anaphase 1, spindle fibers from the poles attach to chromosomes and pull them to opposite ends of the cell. During telophase 1, the spindles and the spindle fibers disintegrate. Then, the chromosomes typically uncoil and new membranes form. This stage marks the end of meiosis 1. Prophase 2 is the first stage of meiosis 2. Meiosis 2 takes place in order to separate the sister chromatids. First, the DNA condenses so the chromosomes are once again visible. New spindle fibers are also produced. During metaphase 2, the nuclear membranes disintegrate. The chromosomes line up on the equator of the cell. This occurs in no special order, also known as random orientation. Then, spindle fibers from opposite poles of the cell attach to each of the sister chromatids at the centromeres. During anaphase 2, the centromeres of each chromosome are split. This releases each sister chromatid as an individual chromosome. The spindle fibers then pull the chromatids towards opposite ends of the cell. Telophase 2 is the final stage of meiosis. In this phase, chromosomes unwind their strands of DNA. Nuclear membranes form around each of the daughter cells, preparing them for cytokinesis. Cytokinesis occurs and the cell division is complete.